everyone, I'm Nadine, this is Yanlin, and we're two AP Bio seniors in Miss Lina's class. We decided to put together a little video or a little interactive lesson about testing COVID-19. So we're going to start by talking about the biological mechanism of the COVID-19 testing kit.
So this is an overview of the testing procedure. We're going to start by taking the sample by swabbing and then adding it to the testing kit. And through some chemical reactions, we get the final sample and we test the content of that sample to get the result. And we will talk about the details of these processes. The first step is swabbing for samples. So specifically for COVID-19, we're using the nasal swab. So why do we get samples from your throat or your nose? First of all, this procedure is painless and it's very convenient for the doctors to perform. And then also on the lining of your nose and the part like connecting to the throat, the cells contain a lot of your genetic materials, which is the DNA. And also specifically for COVID-19, this virus affect mostly your respiratory system, which include your nose, your lungs, and so that's why the doctors take samples directly from your nose. The second step is adding samples into the testing kits. So what are in these testing kits? So first of all is the buffer solution. This is the simplest ingredient, which is just to keep the sample stable. And the second ingredient is the DNA primers for specific viral genes. So when viruses infect your body, they attack your cells and put their own genetic materials into your cells. So for COVID-19, they have RNAs. And when these RNAs gets into your cell, they will become part of your DNA. So what we're trying to do is to use the primers specific for COVID-19 to attach to the infected part of your own DNA, and then we start to amplify these samples. So what we need to amplify for next, we have the primer, which is the starter, and then we have to build on to the DNA. So we need the polymerase and also the DNA nucleotides. The polymerase is an enzyme, which is an chemical just to help carry on the reaction. And we build each nucleotide onto the DNAs, and then we have a lot of samples. And the final ingredient is the fluorescence probe, which is a kind of coloring we use to mark these samples and which contain the viral RNAs and DNAs.
So as Yana mentioned earlier, there's lots of different ingredients in this mixture. And let's just say that there's a little analogy I'd like to make to explain something to you. You have a certain document in a room, and this room is locked. You can't take anything out, but you need what's written on that document to be copied into more components, so into different pieces of paper. And again, how are you supposed to copy that when all of these documents are sealed in this locked room? Think of these documents as DNA. They're stuck in the nucleus and they can't leave. But you need that genetic material to be translated into new pieces of DNA called cDNAs. There's an intermediate step, which is basically the mRNAs. Think of these as kind of like a photocopied version of the DNA in your nucleus that carries the genetic material from the DNA to be able for you to create the new type of DNA containing the same genetic material. This is how it works. You have an enzyme called reverse transcriptase, which is added to your cell mixture that's taken from the swab. This enzyme makes the first DNA strands using the mRNA template. And then the mRNA template is not really useful for us anymore. So it becomes degraded, kind of like the paper shredded. Another enzyme called DNA polymerase, which Yanlin mentioned earlier, synthesizes the second strand of DNA. Now we have a completely new type of DNA that contains the same genetic material. And this type of DNA is called cDNA. Why is cDNA useful? cDNA is useful because it contains the genetic material that we need to test to see whether or not you have the coronavirus. But how can you test that when you only have one strand of cDNA? It's not really possible. which is why we need to replicate the cDNA into billions of the same strand. And that's possible through a process called the polymerase chain reaction, which is a very big kind of like process for something that's quite simple. Now you have one strand of cDNA. Basically, what happens is in 3 different cycles, we're going to end up with billions of the same copy of the same thing. In the first cycle, there's 3 steps: denaturation, annealing, and extension, in which basically this piece of cDNA is ripped in half, copied on each side, and then extended. And once this cycle is over, you have the second cycle, where you go from having 2 strands of cDNA into 4 molecules. In the third cycle, you have 4 molecules doubling to become 8. And intuitively, you can only guess that from here it goes from 8 to 16 and it keeps doubling until you have billions.
So the last step is to mark these final replicated DNAs. So what we use are the fluorescence, which I mentioned before. So what is fluorescence? They're basically coloring chemicals that look like what is in the picture on this slide. They're these neon colored dyes that can mark our viral DNAs. So if these DNAs are not colored, we can't see them under microscope. So there's no way we can test how many of these DNA a person can have in their cells. So adding these fluorescence, we can make the viral DNA visible, and we can actually quantify the strength of and the amount of viruses infected in this person's sample. And finally, we're gonna put the processed sample into the fluorometer, which is a machine that measures the strength of fluorescence. And this will give us different kinds of outcomes. And so let's see what these outcomes mean. So usually scientists look for 4 parts of the RNA from the COVID-19 viruses. So if there are 2 and more out of these 4 types of these RNA strands, we will have a positive result, meaning that that person is actually infected by this virus. But if we get only 1 out of these 4, we will have an inconclusive result, meaning that we're not sure if this person is infected by COVID-19 or is not infected. And finally, if we have 0 out of these 4, we will have a negative result, meaning that this person is clear from this virus, he is not infected.
Moving on to the second part of this presentation, which involves the social impacts of COVID-19 testing in 2 different case studies, the first of which is Jordan. Now, sampling or testing is issued freely by the government, but not for everyone, only for places that are kind of like risk regions. Risk areas are tested automatically and freely and then shut down if tested positive. And only certain people are allowed to leave their homes. For example, people that want to visit banks, that need to go to banks to pay their taxes, people who need to go to the hospital or are leaving the hospital, people who have certain causes of death or cancer, pregnancy, and a few other things. And this is really good because when only certain people have the license to be able to leave the house, that means that we are following social distancing and we are all quarantining and staying safe in our homes. And that reduces the risk of the coronavirus being spread from one person to another, because although it's not an airborne virus, which we're thankful for, it is easily contracted from one person to another. That's another reason why grocery home deliveries and medical home deliveries by the Jordanian government are really helpful for us, because we don't have to go to overly crowded or populated areas to be able to get the things that we need. And otherwise, this is getting some great solidarity for Jordan. I mean, look at this picture on the right, which is a Facebook post from the Jordanian government that gives us daily updates on the amount of cases that go up, that go down, the different hospitals that are monitoring the situation, and how the country is working to deal with the situation. Remember a few weeks ago when we all went on our balconies or opened our windows to clap for the Jordanian community that's all working really hard to make sure this is going well? That's an example of how the country is just kind of like going up with this amazing solidarity as a result of the virus.
So the second case study we're going to talk about is China. China is the place where this virus outbreak started, and it's also my hometown, so I'm going to talk about it. So the policies China has for now for testing coronavirus is being pretty effective. However, even though this is an effective policy, it has its own pros and cons. For the pros is that the government covers most of the costs for testing and treatment if the patient has medical insurance. And also, they're performing mandatory testing for all the travelers to prevent people from bringing the virus in from other countries. And also, they created an online health condition update system for citizens to update their own health conditions. Even though they have health problems unrelated to coronavirus, they can also update through this portal so they can get help from the government. On the other side, there are some cons about these policies. First of all, the accessibility is pretty limited actually, because the applications for free testing requires Wi-Fi and also medical insurance. So people living in very isolated areas like villages in the mountains and maybe somewhere like very far away, they don't have Wi-Fi, then it's hard for them to apply for these free testing kits. And also, they also need to pay for their medical insurance annually. However, some people are in extreme poverty, so they don't actually have enough money to buy medical insurance. Therefore, they can't have the chance to use the free testing kits. And also, there's a very large crowd in China. So even though you're qualified for actually applying for these free kits, you may not get it as soon as possible, and this have been causing a lot of death. And there's another problem with the huge demand is that the production process can be really problematic. So basically, the quality of the testing kits are not consistent. So some people may get very good quality testing kits and get the accurate results, but some people may not. And this is also causing a lot of problems. And finally, these policies are causing like a very potential or long-term negative effect on the country's economy because the government is paying so much for the people. So they need to borrow money from other places to cover these costs. And so in the long term, they will accumulate a lot of debts and they will have to pay back a lot of money. And we only talked about two countries because of the time limit. But if you're interested in other countries, we also gathered some sources and discussion questions to help you guys think about these issues. And I hope you guys enjoy this presentation. And that's it. Thank you.
Thank you so much.